Very good evening, everybody. So I will like to introduce our next speaker, Dr. Ashish Vijay Bakshi. Welcome, sir. Dr. Ashish Bakshi is a medical oncologist at Dr. L.H. Hiranandani Hospital, Mumbai, having more than 3 decades of clinical experience. He completed his MBBS and MD from Nagpur University and DM Oncology from Tata Memorial Hospital, Mumbai. His area of expertise are chemotherapy of solid tumors, head and neck tumors, melanoma treatment, and chemotherapy of hematological malignancies. He also has many publications in national and international journals. I now invite Dr. Bakshi for his talk. Welcome, sir.
Thank you, Dr. Anisha. So in my talk, actually today, I am going to talk about the overview of how the epigenetic machinery can be used to target cancer. So as we have learned from Dr. Mithu's talk, epigenetics is a discipline that studies the reversible heritable changes in the gene expression with no structural modifications in the DNA sequence. So in this epigenetic machinery, as Dr. Mithu has told us, that there are primarily 4 events that happen. One is DNA methylation, histone modification, then there are non-coding RNAs and nucleosome remodeling. And this machinery is often disrupted in malignancies, and these approaches have been used in order to treat cancer also. So in DNA methylation, the DNA could be demethylated, it could be hypomethylated or hypermethylated. Whereas in histone modification, as we have seen in Dr. Mithu's talk, it could be methylated, acetylated, phosphorylated, sumoylated, ubiquitination or polyadiporibosylation, and there are many other histone modifications that can potentially happen. And chromatin alterations can also happen in the epigenetic modifications machinery. This histone modification is an extremely complex process, and as Dr. Mithu mentioned, that there are different histones like H2A, H2B, H3, H4, and all of these modifications can actually potentially determine whether the genes are expressed or repressed. And again, just to recap what Dr. Mithu has mentioned, there are writers like DNA methyltransferase, lysine methyltransferases. Then there are erasers like lysine demethylases, histone deacetylases, and there are readers like chromodomain and bromodomain proteins. And this machinery is used for expression or repression of genes. Now, coming to the cancer-specific part of it, DNA methylation, altered methylation is observed in several malignancies, including the commonly seen ones are breast, colon, esophageal, lung, pancreas, ovary, and prostate cancers. Hypermethylation often results in silencing of the tumor suppressor genes, whereas hypomethylation results in re-expression of the silent genes and genomic instability. Now, acetylation and deacetylation, as has been mentioned, there are enzymes called as histone acetyltransferases and histone deacetylases. So, acetylation results in a condensed chromatin structure leading to cell transcription, whereas deacetylation leads to relaxed chromatin, causing suppression of the gene transcription. So, imbalances in acetylation have been documented in glioblastomas, lung cancers, and AML. Now, there are almost 18 types of histone deacetylases. Now, it is very important to know that which subtype of histone deacetylase is altered in a particular malignancy. Like, say, for example, rise in HDA1C is seen in gastric cancer. Histone deacetylase 5 and 10 are reduced in lung cancer. HDAC1 is increased in prostate and esophageal cancers, and so on. So, it is very important to know it's just not the histone deacetylase enzyme, but what subtype of the histone deacetylase enzyme is acetylated or deacetylated in a particular epigenetic machinery. Basically, lysine methylation can lead to either transcription activation or repression, and it is extremely important to know that which histone is modified and what protein is modified. So lysine at K4 can be monomethylated or trimethylated. If it is monomethylated, it can result in an enhancer activity. Whereas if it is trimethylated, it can result in a promoter activity. Whereas lysine at K9 results in heterochromatin silencing, whereas lysine modification at K27 results in polycomb repression. These are repressive modifications, whereas the red ones that you can see here, these are activation modifications. Now, phosphorylation again is a very, very common cellular process, and it can lead to a lot of changes in energy metabolism, morphological changes in the cells, protein synthesis, gene expression, and a lot of tyrosine kinase inhibitors that are in clinical use. In fact, look at targeting these phosphorylation pathways or proteins. Ubiquitination is another cellular process. Actually, ubiquitin, there are three main enzymes. There are ubiquitin activating enzymes, which are E1. Binding enzymes are called as E2, and ligases are E3. And then there are like erasers in this thing. There are degrading enzymes in the ubiquitin pathway. Diminished activity of E3 ubiquitin ligase is seen in cancers like RCC and breast cancer. Increase in ubiquitination is seen in cervical cancer. Again, elimination of ubiquitination is seen in colon cancer and glioblastoma. Now, these are alterations are extremely important to know, because then you can determine that which specific pathway to target in a particular malignancy. Similarly, non-coding RNA like the small nuclear RNA and small interfering RNA can be used, and this is again a different field altogether of small interfering RNA, which are being exploited in cancer therapeutics. Now, coming to the epigenetic therapies that are being used in order to target this epigenetic machinery. Now, a variety of small molecular inhibitors have been developed in order to target this epigenetic machinery. The DNA methyltransferase inhibitors, as Shyam sir has just mentioned about azacitidine. So there are nucleoside-based drugs called as azacitidine and decitabine. These are commonly used in clinical practice. They are standard of care in the management of myelodysplastic syndrome. I am not going to touch upon them because Dr. Atul Batra is going to talk on them. There are non-nucleoside-based analogs called as hydrolysine, valproic acid, and disulfiram. Particularly, valproic acid is an anticonvulsant, and there are a lot of studies targeting valproic acid in combination with other drugs for the treatment of variety of malignancies. A lot of preclinical and clinical work has been done on this. Histone deacetylase inhibitors are another class of drugs. Again, out of which SAHA, belunostat, panobinostat, these are SGAC inhibitors which have been FDA approved. Panobinostat is approved for the treatment of multiple myeloma. Belunostat is used for the treatment of peripheral T-cell lymphoma. Then aminoanilides like entinostat, bocetinostat, chidamide, these are being used, out of which chidamide is approved by Chinese FDA for the treatment of T-cell lymphomas. Then romidepsin is another FDA approved drug for the treatment of T-cell lymphomas. All of these drugs, they work by working as the histone deacetylase inhibition. So basically they inhibit the acetylation. Then another class of drugs are called as DOT1L inhibitors. This is again part of the epigenetic machinery. This stands for disruptor of telomerase silencing, and a drug called as pinemetostat has been used in clinical trials. Although the results have not been very encouraging, different new classes of DOT1 inhibitors are being developed. Then the antipsychotic drug trinylcypromine is lysine-specific demethylase inhibitors. There are plenty of LSD inhibitors have been developed, out of which many of them are in preclinical and clinical trials. Due to short of time, I'm not going paucity of time. I'm not going into the details of this, but it is important to know that these drugs are being developed, and we will likely to see them in clinical practice very soon. Then out of the repressor complex, the EZH2 is an enzyme that is part of the polycomb repressor complex. And there are two inhibitors, tazemetostat and valemetostat. Tazemetostat is the EZH2 inhibitor, and valemetostat is the inhibitor of both EZ1 and EZH2. Again, Shyam Agrawal sir is going to talk about this, so I'm not going into the details of this. Then bromodomain inhibitors are again being developed. So there is a drug called as birabresib. Then methyltransferase inhibitors for the protein are also again being developed. So the point is that for the different enzymatic machinery or the genetic machinery, epigenetic machinery, all these class of drugs are being currently developed, and 7 of them are currently FDA approved for treatment. So briefly, I'm going to just tell you, Atul will probably discuss in more greater detail about this. But as I said, azacitidine is FDA approved for MDS. More recently, a oral form of azacitidine has been developed and it is available in India also. This particular study, it showed that oral azacitidine can be given as a maintenance therapy after treatment of AML. After acute myeloid leukemia, patients have achieved a complete remission. And this showed that azacitidine reduced the mortality risk by 30% and it reduced the risk of relapse by 40% in patients with AML. So the important message here is that this epigenetic machinery can be targeted with simple oral drugs and which can result in a substantial improvement in the outcomes of patient. These drugs are currently being explored as post transplant maintenance treatment strategies also. Now, azacitidine also has been used in immunoblastic T cell lymphoma. As you can see, a lot of patients given azacitidine for 7 days have achieved almost in 50 to 60% of the patients. Complete response was achieved, and durable long-term remission has also been noted, almost lasting up to 5 years. In this are difficult to treat because otherwise these diseases are very aggressive to treat. Now, this oral azacitidine is being combined with CHOP chemotherapy. So there is a trial called as AzaCHOP. Again, this is being used in peripheral T cell lymphoma, as you can see here. And a remission rate of almost up to 76% is noted in this at the end of treatment. And almost 76% of the patients have achieved a complete response. So these are unheard of responses because typically with CHOP alone, we see our responses up to 50% or 60% like that. So definitely addition of azacitidine is helping here. Then other drugs that I mentioned, HDAC inhibitors like romidepsin and belinostat, they have single agent activity in mature T cell lymphomas. As you can see, almost 30% responses are seen with romidepsin and belinostat, and complete remission rate up to the tune of 10 to 15%. These are again very difficult to treat lymphomas, and in with azacitidine, as I mentioned, angioimmunoblastic T cell lymphoma, you see excellent responses with azacitidine. Now, these drugs are also being combined. So as you can see here, a combination of azacitidine with romidepsin. So a DNA methyltransferase inhibitor with a HDAC inhibitor results in even better responses. So like this combination of azacitidine with romidepsin, it has shown an overall response rate of 73% with a complete remission rate of 55% in T cell lymphomas. So these are excellent and very encouraging results. And phase 3 clinical trials of these combinations are currently underway. Now, these epigenetic drugs are like the chidamide, as I mentioned, it's a HDAC inhibitor. So this drug is being combined with immunotherapy. Sintilimab is a anti-PD-L1 drug. And in patients with extranodal T cell lymphomas, as you can see, almost 50 to 60% of the patients have shown significant responses with the combination of chidamide plus sintilimab. And as you can see, quite a bit of durable remissions are noted with this combination. So again, this is very, very encouraging because these are very aggressive lymphomas and which otherwise really do not respond very well to chemotherapy. Now, as I mentioned, combinations of these drugs are being developed, and the multiple small molecule kinase inhibitors, there are people are doing modification of the drugs so that in single drug you will have dual inhibition of say HDAC with HSP90, HDAC with DNMT, HDAC with EZH2, and so on. So a lot of multi-targeting agents are currently being developed. We won't be going into the details of this for the paucity of time. Another unique type of strategy that is being used to target the epigenetic machinery is called as PROTACs, which stands for proteolysis targeting chimeras. Now, basically what happens is that the ubiquitination pathway is responsible for the degradation of the proteins that are not required by the body. So what in PROTAC, what is done is that you have a protein, we have a ligand, which is for a target protein, and there is a linker molecule, and there is one ligand for the E3 ligase. So say, for example, this particular protein we have to degrade. It is here, it is this like ligand will get attached to this target protein here, whereas the E3 ligase, so this protein will be ubiquitinated and by the E3 ligase, and eventually there is a trimer formation of the drug, the linker, and the E3 ligase. Now, multiple ubiquitination tags will keep on happening in this protein, and eventually this protein will be marked to the proteasome machinery, where the proteasome machinery will degrade this protein and bring about the destruction of the unwanted protein. Now, how this protein is protag is being exposed in epigenetic therapy. Now, prostate cancers are androgen dependent cancers, and in one study, a molecule has been drug called as ARV-110, and this degrades many common form of the androgen receptor forms. Now, in this phase 1 study, what they've shown is that those patients who had mutations in this location of the androgen receptor, they showed more than 50% reduction in the PSA of the patients. So this drug, that ARV-110, is now called as bavdegalutamide. And in a phase 2 study, they used multiple patients who had androgen receptor sensitive mutations. And these AR672 and ARV7 variants are resistant to the bavdegalutamide. Now, in this overall PSA, 50% reduction of PSA was seen with this drug in 15% of the patients, and 30% reduction was seen in 30% of the patients. But in the sensitive androgen receptor variants, that is the 878A and the 875Y, there was a 50% decline in PSA in almost up to 50% of the patients. This is an oral drug. So this protag is an oral drug, and this can be given to the patient. And this is a group of patients who have received two androgen receptor targeting drugs. So even in these highly treated patients, there is a 50% reduction in the PSA, which is a significant response. So probably this drug will move to a early setting, and we are looking forward to this drug with a great enthusiasm. Now, another therapy that is being used for targeting the epigenetic machinery is called as the Crispr epi editors. So the Crispr is a gene editing technique. So CRISPR stands for clustered randomly interspersed short palindromic repeats. In this, basically, RNA is marks the site where the DNA needs to be pricked, and this Cas9 is a cutting device. So once the CRISPR's RNA recognizes and binds to the target DNA, this Cas9 enzyme will cleave that DNA part, and then you can insert a new genetic material here in this. So how this technique of CRISPR gene editing is currently being explored in a lot of hereditary conditions, including thalassemia. How this is being exploited for the treatment of cancer. Now, what people have done is that they have taken the patient's T cell. There is a specific gene called as NY-ESO1 receptor. Now, these CRISPR edited T cells are made in such a way that this NY-ESO1 protein is inserted, and these three proteins called as PDC1, TRAC, and TRBC are deleted. And then once this modification is done in the T cell, this is a CRISPR edited T cell, you multiply this T cell, and then you infuse these T cells to the patient, and eventually these modified T cells will go and bring in the killing of the cell. Now, a phase 1 trial of this particular CRISPR edited T cells has been done, and reductions in the melanoma patients have been noted. So currently, not all patients responded to this technique. So a lot of modification needs to be done in this technique, and trials are underway to make this treatment successful. Now, this is a very, very interesting technique, because by using the CRISPR epi editing technique, you can bring about transcriptional regulation. So basically, you can insert an activator or a repressor. You can do a histone modification by inserting the demethylases or methyltransferases, histone acetyltransferase, and so on. You can bring about DNA methylation by activating or repressing the DNMT or TED, and you can even target the chromatin with the help of a fluorescent probe. So like this, the CRISPR technique can be used for a variety of purposes, and these are currently being explored for the treatment. Now, also the epigenetic machinery is involved in the development of chemotherapy resistance, because when the patients are receiving chemotherapy, there are a lot of abnormalities in cell energy metabolism. Then P-glycoprotein efflux pumps and all that machinery is also deregulated. Then there is DNA methylation and histone modification results in apoptotic tolerance and autophagy. Then increased tumor cell signaling and so many other abnormalities happen. There are alterations in the fibroblast, hypoxia and defect factor, and so on. So in short, a lot of epigenetic mechanisms are responsible for the development of resistance to chemotherapy. So what scientists are trying to do that now is that they are trying to use a drug targeting the epigenetic machinery in order to sensitize the cells to the chemotherapy. So one example of this is this study published by Tina Badla. This was way back in 2012, where they have shown that epigenetic reprogramming reverses the relapse-specific gene expression signature and restores chemosensitivity in ALL. So as you can see here, untreated cells, a lot of genes are activated here and a lot of genes are repressed here. And in the group of patients that were treated with vorinostat, there is a change in the alterations of the genes. So this has shown that actually epigenetic machinery can be targeted in order to increase the sensitivity of the chemotherapy. Now, one example of this is also that epigenetic therapy is being used in order to increase the responses to immunotherapy. So as I mentioned previously, epigenetic machinery is responsible for a lot of immune dysregulation. So in this one interesting study published at ASCO 2020, just 2 years back, what they have done is that in a group of patients, they used immunotherapy called as pembrolizumab, whereas the other arm received a combination of pembrolizumab with a vorinostat, which is a HDAC inhibitor. Now, as you can see here in arm way, which was pembrolizumab alone, you can see partial responses of 25% and disease control rate of 58%. As against that, in the group that received vorinostat along with pembrolizumab, you see partial responses of almost 50% and a disease control rate of 91%. So this is a very, very interesting proof of principle study. We showed that by combining epigenetic machinery with immunotherapy, you can have more depth of response and also more durable responses. So a lot of trials are currently trying to address this issue. So to conclude my talk, epigenetics is a very exciting and rapidly evolving branch. Alterations in the epigenetic machinery are responsible for the development of several cancers. The epigenetic machinery can be targeted by a variety of mechanisms, including small molecule inhibitors, PROTACs, CRISPR-based gene editing, and RNA therapeutics. Significant responses have been obtained in relapse refractory malignancies, as I presented in my talk. Combinations of these drugs with traditional chemotherapy, immunotherapy, and other targeted agents are currently being exploited, and a discovery of new molecules is expected to revolutionize this field. So with this, I would conclude my talk, and if there are any questions and questions, I would be glad to answer them.
Thank you so much, sir, for your very informative talk. In the QA right now, we have no questions, sir. So we'll move ahead. By chance, if any questions come in between, so we'll let you know. Thank you so much.
Thank you.
Thank you so much, sir.